So it's my pleasure and honor and pleasure again to introduce Professor Paul Cumming, who is our constant collaborator, who is incredible professional in the field of brain imaging and dopamine. Professor Paul Cumming is working in the Department of Nuclear Medicine in the University of Bern in Switzerland. Also, Professor Paul Cumming is adjunct professor of School of Psychology and Counselling, Faculty of Health in the Queensland University of Technology in Brisbane, Western Australia. And we are very happy to have Paul in our team as a visiting professor and chief researcher in the International Center for Education and Research in Neuropsychiatry, Samara State Medical University. Paul will discover the topic of molecular brain imaging of Huntington's disease today. Thank you very much for being with us, dear Professor Paul Cumming.
Well, thank you. I'm very glad to be here, although I'm not sure where here is, since this is a virtual meeting, and I guess we're rather in the manner of electrons, we're non-localized. We could be anywhere. But again, I'm going to pick up from where Professor Svetlana left off, which is kind of starting from, she really reviewed how Huntington's disease is a systemic disease, which is something that I had been not so aware of until recently, having focused in my readings about Huntington's on the central nervous system manifestations. But it is very importantly a systemic disease, and indeed, as we've heard, the major important causes of mortality arise from its peripheral manifestations. And in that respect, I see a resemblance with Parkinson's disease, which is also a systemic disorder, although that's less appreciative because the peripheral manifestations such as autonomic dysfunction and gastrointestinal difficulties, constipation, for example, are less life-threatening in Parkinson's, but it also bespeaks of the widespread manifestations of the disease, whereas the principal neurological symptoms are attributed mainly to one particular locus involving the basal ganglia. And this too will come up in my little review of brain imaging of Huntington's disease. So molecular brain imaging refers to the relatively new technologies that's developed over the past 30 or 40 years to visualize specific molecular entities in the nervous system. And since this procedure or this technique is not so familiar to some of you, I'll give a quick review of that. Next slide, please. I forgot about that slide. So functional imaging or molecular imaging of molecules is distinct from the structural imaging that can be attained with magnetic resonance imaging, or indeed post-mortem examination, as in this example, where on the left side we see a normal, healthy, you would say, slice of a healthy cerebral hemisphere, and on the right hand, a clearly atrophied brain that came from a patient who died with Huntington's, I presume at a rather advanced state. You will see that there is, just from visual inspection, that there's cortical thinning throughout due to global atrophy and associated loss of white matter, most notably the basal ganglia, which is present in the well-developed caudate, putamen, and indeed the nucleus accumbens, seen in the healthy left side brain, is nearly completely lost due to the degeneration. And that process is indicative of rather selective, but not exclusively selective, neuropathology of Huntington's, in which the extended striatum, the dopamine-rich part of the brain, is somehow particularly vulnerable to the pathology of Huntington's. And furthermore, the movement disorders, at least aspect of Huntington's, is clearly related to the degenerative changes in the basal ganglia. Next slide, please. Oops, we skipped over one. So that little instrument there is, this one here. It's a photograph of the cyclotron, the very first one that was built by C. Lawrence, who got the Nobel Prize for that discovery. And you can see that in the 1930s, physicists still worked with brass and sealing wax and made and blew glass structures themselves in the lab. And he made this cyclotron. You can see the two so-called Ds. It consists of segments in which there's an oscillating electric potential applied. And inside there would be, in its normal operation, a vacuum. And placed between a fixed magnet, this causes particles in the cyclotron to accelerate as they spiral outwards. And in the next slide, yeah, that's what I meant to show in this order. This is a modern cyclotron. It really shows the same principle, but this is a medical cyclotron specifically used for accelerating particles, protons in general, to generate synthetic radionuclides to be used for molecular imaging, such as PET. And you see, it's not that unlike the Lawrence's instrument, except it's much bigger, but it has these copper segments inside that apply the oscillating electric field. And you see around the instrument, there are exit channels or targets, which are really very much like in Lawrence's prototype from 1937, I think it was. Those targets would be, if we go back to the previous slide, would be where the actual alchemy of really transformation of nuclei takes place, where the high energy beam of protons indicated by P in some of these nuclear reactions shown here are thrown at enormously high velocity at a target to produce, let's consider just the top one, for example, nitrogen, natural nitrogen of the naturally occurring gas, when hit with sufficient energy by protons, will undergo a nuclear reaction to produce a new nucleus called carbon-11, which is a very short-lived radionuclide that can be incorporated into, you see the upper right-hand arrow leading to carbon-11 H3I, that is to say methyl iodide, which is a synthon or a reactant that can be used to integrate that very short-lived radionuclide into any number of molecules which are prepared to react with methyl iodide, which is a reactive nucleophilic reagent. And so there's a huge spectrum of possible molecules that can be labeled with either carbon-11 or, in the lower portion of the figure, fluorine-18. Carbon-11 has certain advantages, but it's logistically difficult to use in PET imaging because it has a physical half-life of only 20 minutes. That means you have to fire up the cyclotron and really within a matter of half an hour produce the carbon-11 and do some very rapid chemistry that has to be up to GMP practice to produce a product within less than an hour that is ready for intravenous injection into a volunteer. And this is not something that can be done everywhere. It requires a lot of coordination. The lower part of the figure shows the other widely used medical radionuclide for molecular imaging, which is fluorine-18, which is likewise produced in the cyclotron, but with different targets and different energies involved, to produce fluorine-18, which has a half-life of 2 hours. And that is much more convenient because you can make it in the morning and even ship the product some distance, sometimes hundreds of kilometers away to the point of use. But the important difference is a chemical one, that whereas many biomolecules and medicinal compounds normally contain a methyl group that could be amenable to labeling with carbon-11 methyl iodide, it's a relatively smaller range of pharmaceuticals that normally contain fluorine But fluorine chemistry has developed in leaps and bounds so that there are now many different compounds that can be labeled with F18 for PET imaging. Next slide. Next slide, skip over that one. That was out of order. So here, if you click again, you'll see the principle of positron annihilation. Advance the slide. So there you see a proton, and rather a nucleus on the right-hand side occurring in the medium of the brain, and a ligand, that is the molecule that contains, let's say, fluorine 18, and decays with a half-life of 2 hours, randomly releasing a antimatter electron known as a positron, beta plus, which floats around in the medium of the brain, or rather it travels with some high energy. Next, forward. And it then encounters the electron in the medium of the brain, positive and negative charges being the only difference between the positron and the electron, but they are attracted to each other electrostatically. Next slide. And they merge together in this fatal attraction as a short-lived element, let's say, called positronium. Next slide. Which immediately annihilates, not immediately, almost immediately annihilates, releasing all the energy bound up in that electron-positron pair as two photons. You can see one leaving from the top and one leaving from the bottom as waves. Now that energy is very strictly defined by the famous equation E equals mc squared. And as it turns out, the energy of those two particles, those two photons leaving the site of annihilation, is equal to the rest mass energy of the electron-positron pair, 512 kilo-electron volts. Next slide. Now that is a high energy gamma ray, and that will pass through a centimeter of lead, and it'll certainly pass through your head. Here we see, I'm not sure where this slide comes from, but it's one of the very first prototype PET scanners when, in the days when people wore them like a hat. And you can see that there are only about 10 detectors around the array, but the principle of detection of PET relies upon the simultaneous emission of the two gamma rays pretty much at 180 degrees from each other. And if they encounter two detectors separated across this ring, which would take some luck because these are relatively small detectors in that prototype scanner. But if it does happen that two events are registered by the detectors on either side of the ring in the same instant, that counts as a decay event. And that registers towards eventually producing an image. Next slide. And that positron annihilation here taking place inside the gantry of a relatively recent PET scanner, the one that they were using at UCLA about 15 years ago. That event takes place in, imagine the patient or the volunteer lying in the scanner with their head in the hidden rings. Notice that modern scanners hide the wires nicely. That's a little bit of a joke, but inside that scanner, there are thousands of wires connecting all of the different detectors hidden inside the nice white chassis of the medical instrument. Because each pair of detectors separated across that detector ring have to be wired to each other so that some synchronous events of gamma detection can be registered. Next slide, or forward. Sorry about the clicking noise. So here you see again the emission of two gamma rays at nearly 180 degrees. Ahead, next slide. And then you see hidden behind the white chassis of the scanner, there are rings of detectors, and here two of them are linked together and they happen to register that particular decay event. Next slide. There is a ring detector, actually, modern scanners have many rings extending over very large fields of view, all wired together in a nightmarishly complex and expensive piece of apparatus. Next slide, forward. And so you see the kinds of rays that are possible for different decay events arising when two detectors, any possible pair of detectors in the ring, are activated in the same moment. Next. And these are parallel rays. Go ahead, next slide. And so by counting millions of those events, millions, literally, and applying some algorithms for reconstructing what the source looked like, that's a matter of medical physics, you can produce an image of the a source map of the brain showing where the radioactivity came from and how much there was at any time. Now, I happen to know, or anyone who knows much about PET will look at that and say, it looks like an FDG PET scan, and in fact it is. Fluorodeoxyglucose is the most widely used tracer for molecular imaging in brain because it's so darn useful. Fluorodeoxyglucose contains fluorine 18 in its nuclear medicine application, and otherwise contains a derivative of glucose that behaves much like natural glucose, but carries with it the radioactivity from the fluorine 18. And so it allows you to track the metabolic activity in brain. Next slide. And in very formal terms, we can represent that as a series of forward and backward reactions where the fluorodeoxyglucose, which I call the best PET tracer because nothing has worked quite so well and so usefully as fluorodeoxyglucose, which we call FDG. Now, the tracer is administered by an intravenous injection. And so beginning on the left side of this figure, we see that FDG is present in the plasma of the blood and circulates along with the blood. And when it passes through the brain, the radioactivity in blood is able to transfer across the blood-brain barrier. This is a reversible process. It's not simple diffusion, but it's mediated by a transporter in the blood-brain barrier. So that some like natural glucose, FDG ends up in the brain tissue. And there, it can be phosphorylated by the enzyme hexokinase in living cells. And unlike natural glucose, which continues along the path to ultimately to the Krebs cycle and is exhaled as carbon dioxide, fluorodeoxyglucose gets stuck. It gets metabolically trapped. And so it continues to pile up and pile up and pile up and accumulate in brain and in any metabolically active tissue that would normally be burning or consuming glucose. And so that is the fundamental basis of the PET signal specifically with fluorodeoxyglucose or FDG, that it's trapping in a metabolically active tissue like brain, and there's no tissue so more metabolically active than brain. It gives you an index of how rapid the metabolic rate is in that tissue, and that can change from day to day, or it can change as you age, or very importantly, certain neurodegenerative diseases will compromise the normal metabolic rate in brain. Next slide. And that actually, FDG was being one of the very first, and as I said repeatedly, still the most successful tracer, was used in what we call the early days of PET research. The early days of PET research means the 1980s. And one of the relatively first studies of really in the realm of neurology was the use of FDG to study the very mysterious and catastrophic disorder of Huntington's disease. And this paper reported that really for the first time in a quantitative sense, where they obtained PET scans with FDG glucose in a group of healthy controls and in patients, and published their results in the Annals of Neurology. Next slide. Yeah, and this figure extracted from their paper summarizes the results. And on the left-hand side, we see a table reporting the metabolic rate that they calculated for different brain regions in the left-hand figure of the table, control subjects, and on the right-hand side, the Huntington's patients. And the red bar indicates the point of main interest, that this metabolic rate calculated from dynamic FDG PET recordings is enormously reduced, really by more than half, in the caudate nucleus and in the putamen of the unfortunate patients with Huntington's disease. And that represents a massive loss of metabolic activity. And furthermore, on the right-hand side of the figure, you see that individual results by this quantitative PET method correlated very, very significantly with various measures of functional capacity or severity of the disease. So the more severe, the more advanced the disease in individual cases, the more loss of metabolic activity there was in the caudate and putamen. Equally, caudate and putamen being the main targets. And using the technology that was available in 1980s, they weren't able to detect any changes in FDG glucose metabolism outside of the caudate and putamen. But PET scanners today are much, much better and more sensitive. And I have no doubt that an investigation using modern instrumentation would reveal a more complex pattern of metabolic fallout in Huntington's. Next slide. Cortical atrophic hypometabolic metabolic association in the transition from pre-manifest to early Huntington's disease. Now, I took this paper that was published only a few years ago as a good example of how things have progressed, because it was pretty easy, even in the 1980s, to use the PET to detect a massive metabolic loss in defect in the caudate and putamen of patients with manifest disease. And at that time, there was a period in the early 1980s when before the discovery of the Huntington gene, I think in 1983, if I'm not mistaken, there was a brief window of time where FDG PET could have been used to detect preclinical loss of metabolic capacity in the caudate putamen in people who had a family history of Huntington's and did not know and worried that they might be at risk for developing Huntington's. And I remember discussing with, I was a graduate student at the time, but I remember discussing with a neurologist how that created sort of an ethical problem, because genetic testing was not yet available. And yet there was a period when, lasting only a few years, where FDG PET could have in principle been used to detect the pre-morbid or preclinical manifestations of the disease, which presented some conundrums for those with a family history of the disease. Now, of course, this is not necessary because anyone can get screened genetically to see if they have the CAG repeat mutation of Huntington's disease. But it's still a problem, it's still an interesting question about when the disease really starts. And can you detect, you use PET to detect changes in metabolism that might precede the onset of the disease, or distinguish between the preclinical state and an actual clinically manifest Huntington's. Next slide. And here, it's a little bit complicated because I told you at length about FDG glucose, but here they have also availed themselves of multimodal imaging using MR scanning to measure in the same subjects the cortical thickness. And if you remember from the gross anatomic slide at the beginning, cortical thickness atrophy is clearly indicated in the end stage of the disease. But it's also occurring as the disease progresses. And here they compared, on one hand, MR-based cortical thickness measurements, detected, I think these are called zebra images, where they have, in effect, projected a shaved brain that has been trimmed off so you can see the deep matter sulci and the sulci and the white matter, underlying white matter revealed in the same surface rendering of the brain. It's a common problem in all brain imaging, how to represent the complex 3D surface using only the 2 dimensions of the printed page or the computer screen. So here on the red scale, you see that they contrasted very early Huntington's with the very first manifestations compared to people who were known carriers and therefore sadly doomed to develop the disease, but had as yet no actual evident motor disturbances or cognitive disturbances. And in the red scale, you see that there is a cortical thinning in, well, it looks like sort of the prefrontal lobe, that is the premotor cortex, and in some regions of the occipital lobe, or rather in the motor strip, you know, and this kind of makes sense functionally because this is where the disease is ongoing. But interestingly, the FDG PET investigation in the same subjects shows that the loss of metabolic activity in this very early stage of Huntington's is present in quite different regions than where the cortical loss is taking place. In particular, you can see in the blue a very distinct loss of metabolic activity in the frontal cortex, really, and almost nowhere else at this stage of the disease, and in the anterior cingulate cortex. And no doubt this accounts for some of the behavioral manifestations of Huntington's, which might involve disinhibition or impulsivity due to impaired frontal function. But it's interesting that it's physically separated from the points where the actual cortical loss is taking place. So it's probably a kind of a downstream effect of the primary degeneration of the brain at that stage. Something to keep in mind, that metabolic disturbance doesn't necessarily mean, in this case, atrophy, because the metabolic loss in these frontal lobe of these patients precedes the onset of distinct atrophy or tissue loss. Next slide. Next slide, please. Now, it's a very nice pattern for wallpaper or something like that. These are correlation matrices. So rather than considering an individual brain region, here we see FDG PET results in a whole series of subjects. Each one of these matrices is a grid, and it shows the correlation matrix between glucose consumption in 30 or so different regions, 45 regions of the brain, correlated with every other 45 regions. And you see that there are zones where there's a high correlation, it's indicated by red. Never mind what regions they are, the text is far too small to read, but a normal brain shows clusters of brain regions that are interacting with each other and are sort of covarious, so that they have naturally high activity together in parallel, and other regions shown in blue where there's lesser correlation. And so within a series of subjects, healthy subjects, you could get a whole range of results, but the overall pattern is the same, that some regions of healthy brain are sort of talking to each other and are entrained metabolically with each other in the normal operation of the brain. Next slide. And this kind of general approach of considering rather networks instead of discrete areas is very powerful for a tool for investigating the basis of neurodegenerative disorders, where that might be considered as disconnection syndromes, where some parts of the brain that normally should be in communication have fallen out or lost their normal relationship. And David Eidelberg from Long Island, I guess, has been a major proponent of this approach to, in effect, extract out patterns that are characteristic of the metabolic disturbance of one disease or another. And for example, the metabolic pattern that he discerns for Huntington's disease is very distinct from that which is described for Parkinson's disease, despite certain vague similarities in pathologies. Next slide. And the upper part of this figure shows Eidelberg results where in effect, this is a population-based result showing the pattern of abnormal activity in a population of Huntington's as distinct from a healthy control group. And you see that there are regions where the Huntington's patients have characteristically lower activity indicated in blue, notably in the caudated putamen. If you look at the third tile to the right in the upper figure, that's not surprising, but there are also some hypometabolism in the insula and in the prefrontal cortex, unsurprisingly, based on the early results. And there are also some regions where metabolic activity is relatively increased in Huntington's, in the cerebellum and in the pons, shown in red in the left-hand upper figure. So using this kind of network analysis, they were able to interrogate the data and kind of answer the question about how long does pathology precede clinical manifestation? And using patients who were, I guess, genotyped for their number of CAG repeats, which is a clear predictor of how soon they're going to develop the disease. In the middle part, figure C, tile C, never mind the details, but it shows that within individuals who were scanned at, over a period of 5 years at intervals, you can see them tracking or progressing along a line that will bring them into the metabolic pattern of pronounced Huntington's disease. So this is, I think, a very impressive paper because it shows, using a metabolic network approach, that the process leading to disease is already established even 20 years before the clinical manifestation of Huntington's disease. So it's in small increments that the brain loses its normal functioning. And that, in a sense, might be hopeful because it means that although the disease process is already established decades before, and of course in childhood Huntington's it can happen much earlier, but normally the disease declares itself in the 50s, and it might give scope for treatment because it means that if you could interfere with that process, then you could rescue the disease. Next slide. So I've talked only about FDG, but I've pointed out that the one of the very early findings in metabolic PET, in addition to the hypofrontality, is the loss of metabolism in the basal ganglia, which is a vulnerable region in the brain. And the basal ganglia, normally referring to the caudate and putamen, is the dopamine-rich part of the brain that substantially degenerates in Huntington's disease. And as could be seen in that gross anatomic specimen where there was practically none remaining. And that surely is responsible for the main part of the motor symptoms of Huntington's. Now, people then at, actually in Switzerland, where I happen to sit right now, were early investigators of not just the FDG PET, which, as you saw, was already documented in the 1980s, but they also applied dopamine PET using a different tracer, not FDG, but a dopamine marker, to investigate simultaneously in the same subjects, not simultaneously, but in different sessions, on one day FDG PET and another day PET for dopamine receptors, to look at the relationship between the metabolic disturbance and a specific neurochemical marker for the integrity of the dopamine system in the brain. Next slide. And that result is summarized here, showing in the upper figure the metabolic map using FDG. It's hard to tell, but the clinically manifest patient in the upper left-hand corner, yeah, with a little bit of imagination, you can see that there's hypofrontality compared to some asymptomatic gene carriers on the right-hand upper figure. And if I had a pointer, I would point out where the basal ganglia is, because actually the clinically manifest patient has a distinct loss of metabolic activity in the caudate and putamen, although it's a bit hard to see from the figure. Now, looking at the lower part of the figure, you see their corresponding results from the same 3 individuals with the dopamine marker, carbon 11 raclopride, that shows the abundance or availability of dopamine receptors in the caudate and putamen. Of the clinically manifest patient, there's a very substantial reduction in raclopride binding, 50% below what is present in the asymptomatic carriers. And that is indicative of the degeneration of the neurons of the striatum, which bear the dopamine receptors. And in fact, I would go further to say that patient number 1, or rather the asymptomatic carrier number 1, is halfway on the road to Huntington's compared to the right-hand-most person who seems to have a much more intact basal ganglia. So that's interesting because we can see separate channels, energy metabolism on the upper figure and dopamine receptor binding in the lower figure. And both of these changes, degenerations, seem to occur in parallel. Next figure. And here, a more recent application from the same, some of the same people, Nico Leenders, who used to be at the Paul Scher Institute in Switzerland, but has since then been returned to his native Netherlands, and continued that kind of work and much other very interesting work. And this research group here presented 10 years ago the results of a more kind of refined analysis of dopamine receptor binding, again, at that very critical transition between preclinical Huntington's disease among carriers who do not yet manifest conspicuous symptoms. And I bet they do have symptoms if you look closely enough. But this point of transition between the preclinical syndrome and the point where they actually declare symptoms is a very interesting point, because it indicates a threshold for pathology and a threshold where you would have to embark in a treatment before, if you were to rescue these patients. Next slide. And it's numbers, but it shows what a fine point this is. Never mind the units, but binding potential is the standard unit for receptor binding studies by PET. We call it BP, which sounds like blood pressure, but it means in this context binding potential. And if you look very carefully in the upper part of that figure, the putamen of the preclinical carriers who have not yet established manifest symptoms is 2.3 units in the carriers versus 3.0 units or 2.9 units in the controls. So in other words, and it's highly significant. So they don't have symptoms, but they already have a 20% reduction in dopamine receptors in the putamen. And furthermore, to follow up, you see where the red bar is, they went from 2.3 to only 2.1. That is, say, a 10% decline just in a couple of years between scan 1 and scan 2, whereas healthy controls only have a very slow loss of dopamine receptors to follow up. So there's an ongoing process that causes further loss of the dopamine system to follow up, but more importantly, perhaps, this shows unambiguously that even before they manifest in symptoms, carriers who are destined to develop Huntington's already have lost a substantial proportion of their dopamine receptors. Next slide. Now, this is my own figure, just from some rat studies to introduce an important concept in the dopamine system, where we have autoradiograms, or it's like a poor person's PET, where the brain section is incubated with various radioligands to reveal the dopamine receptors in the upper figure. And in the lower part of the figure, you see much the same image, that is to say, dopamine receptor binding, but this is not receptors, this is the dopamine transporters in the lower part of the figure, the cocaine binding sites that are indication of the density of innervation in the brain. And they seem to match. The receptors are present in the striatum, but in the cells resident to the striatum, these are the cells that degenerate in Huntington's. Whereas in the lower part of the figure, you see the dopamine transporters that are present in the dopamine fibers innervating the striatum, pre and postsynaptic. Next figure. So it presents a question we've already seen in the next figure, please. We've already seen that the postsynaptic marker of dopamine receptors undergoes a massive degeneration in Huntington's, and that degeneration precedes the onset of symptoms. But what about the presynaptic marker, the one that degenerates in Parkinson's disease, the actual innervation? And that can be studied with other tracers that target specifically the dopamine fibers, as in this study. Next slide. And it's a little bit hard to see, but they use something called TRODAT, which is a SPECT tracer, not a PET tracer. But it shows very clearly the striatum in a healthy control lighting up, because the dopamine innervation labeled by TRODAT SPECT is completely intact in the control, and varying degrees of degeneration in Huntington's patients. But it's not very pronounced. It's actually rather modest loss compared to the massive dopamine receptor loss. And in fact, Huntington's disease relatively spares the dopamine innervation, but the innervation doesn't have anywhere to go, because it's looking around and it's still finding its target in the striatum, but the dopamine receptive cells are essentially lost. So you have actually too much dopamine in a sense, because the dopamine innervation is intact, but the responsive cells to dopamine have degenerated. Next slide. So actually, how much time do I have? Have I been talking too long? Am I good for another 5 minutes or so?
Dear Paul, we have around 3 minutes because...
Oh, okay, well, I'll have to speed up my tempo. So these are microglia. This is something completely different. This has nothing to do with dopamine or glucose, but these are the resident macrophages in the brain, here in the rat, labeled with a marker for microglia. And these are the inflammatory cells, because Huntington's disease is an inflammatory disease. And if we go to the next slide, we can see that various people, including Bonatti and people from London, were very interested in looking at microglial activation in dementia. Next slide. Using a PET tracer specifically targeting something on the surface of microglia. And you see the bright, in the upper figure, a healthy person, aged 74 years, who was very sparse microglial binding in brain. And then Alzheimer's patients have very abundant binding, showing the microgliosis that is shown schematically in the lower part of the figure, in the temporal lobe and the hippocampus, which are just the regions known to undergo the most degeneration in Alzheimer's disease. Next slide. So one could ask the same question, and they did. The London group investigated using the same tool, the microglial reaction in Huntington's patients. Next slide. Here is shown schematically where in an inflammatory state, an microglia first become activated and then overexpress this marker that is labeled with the carbon 11 in the PET experiment. And then ultimately they can transform into the aggressively phagocytic phenotype of microglia. Next slide. And I've made an error in this slide, but what it shows is in the so-called glass brain depiction, which is another technique for representing 3D data, as if the normal part of the brain were transparent, but the black zones are where it should properly read microglial marker increased in Huntington's disease. I apologize for that error. So you see that the microglial activation is present, if you know how to read these scans, in the basal ganglia and in the frontal lobe, which is just where you would expect the disease to be active. And in the same subjects, the dopamine receptor marker, just as in the previous study by from Switzerland, is also decreased. So in other words, where there is dopamine loss, there is microglial activity, indicating an ongoing inflammatory and destructive process of the brain. Next slide. And we don't know exactly what causes that, but interestingly, these people continued that line of work and considered the relationship between microglial activation in Huntington's disease and peripheral markers of an inflammation, that is cytokine levels in plasma. Next slide. And yeah, it's a little bit hard to read this slide very quickly, but they show that pre-manifest Huntington's disease patients, that is genetically labeled, disposed to get the disease, but not yet manifesting in symptoms, already have substantial microgliosis, as shown in the bar diagram on the upper right. You can just see that the pre-manifest patients have about twice as much microglial activity as the controls. So once again, for me, this is the most important lesson that I drew from putting together this talk, which is that the disease is already well established and underway long before the appearance of symptoms. And in particular, the inflammatory activity of the brain is already established. And furthermore, it's perhaps less interesting, but they showed that interleukin levels in plasma were elevated, suggesting that there's a peripheral component to the inflammatory response also in Huntington's, which is consistent with what we heard from the previous lecture, that Huntington's is a systemic disease involving pathologies in practically every organ of the body. Next slide. And we'll just skip over that, but it shows that there's a correlation between individual plasma markers of inflammation and the PET results for microglial PET. And I think the next slide brings us to the final topic, which I'm going to mention, which is that receptors like dopamine receptors are just part of the immensely complex machinery that instructs and informs the receptive cells to change their metabolism and behavior. And of course, this is meant to convey a sense of how complicated it is, but I won't go through it in detail. But GPCR, G protein coupled receptors, would be dopamine receptors, but there are many other entities on neurons and other cells that are responsive to signals and all converge to activate different pathways in the living cell that are collectively known as second messenger systems, which might converge on the regulation of cyclic AMP, which is the effector molecule in many cells that ultimately controls gene expression, phenotype.
Thank you very much, dear Paul. Thank you.
So is that it?
Yes, dear Paul, so will you summarize?
Well, I overestimated by just a minute, but if we go to the final slide, and that is the final slide. Proceed again. Skip over the next two slides. I apologize for going on too long. Yes, here we are. These people did a very new kind of PET using a ligand for phosphodiesterase, which is a signal transduction molecule. And in the very final slide, please proceed. You can see in the upper figure that as the Huntington's disease progresses, the abundance of this marker of second messenger signal transduction declines inexorably from the control case to progressively disappearing completely in advanced Huntington's disease. And this occurs in parallel with the decline in dopamine receptors. And I believe that is where I stop and where I had planned to stop. But it shows that there's a very broad spectrum of different aspects of neurotransmission that are affected in Huntington's. And my main summary point is that there is a pre-morbid, pre-manifest pathology that can be detected by PET even before the neurological and psychiatric symptoms of Huntington's declare themselves. And really, my last statement would be that unless we come up with a treatment, unless somebody comes up with a treatment or an intervention, Huntington's inexorably proceeds to this very horrible state of the nervous system where really nothing works. And certainly the dopamine system is completely messed up. So at that point, I would stop, and I'll look forward to receiving some questions that I hope I can answer. If I can't answer them, I'll find someone who can. Thank you very much for your attention.
Dear Paul, thank you very much for your lecture. And we will send questions soon after the event, after we finish all the design of the lectures, and we will report about some technical issues. We sincerely apologize in front of the audience that YouTube stream had some difficulties, but we will do our best to prepare these two amazing, brilliant lectures for the web library of neuropsychiatry. Thank you very much, and see you soon. Bye-bye.